every single day, in IVF labs all around the world, embryologists are looking through their microscopes and witnessing this incredibly frustrating mystery.
Oh, absolutely. It's the classic day 3 heartbreak.
Right. You pull a dish out of the incubator on day 2 and you look at the embryo and it is just beautiful. I mean, it's cleaving perfectly, 2 cells, 4 cells, totally symmetrical, zero fragmentation.
You feel great about it. You're ready to freeze it or transfer it.
Yeah, exactly. But then you look again on day 3 and it's just stopped. It's completely arrested at the 6-cell or 8-cell stage. And, for decades, we just blamed poor morphology or bad egg quality.
Which is such a massive oversimplification, right? Because the real culprit isn't something you can actually see with a standard optical microscope. It's invisible.
Right. It's a complete catastrophic failure of the epigenome.
Yeah, that day 3 arrest is honestly the perfect place to start.
Yeah.
Because what you are seeing under the microscopes is basically a biological machine that ran out of its initial battery power.
And it failed to plug itself into the main power grid, essentially.
Yeah. The blueprint of the DNA was sitting right there in the nucleus, totally intact, but the embryo literally just forgot how to read it.
Well, welcome to this deep dive. Today, we are speaking directly to you, the clinical embryologists, specifically those of you who are currently poring over textbooks, getting ready for the massive undertaking of the ESHRE certification examination.
And look, the ESHRE board doesn't want you to know epigenetics just to torture you with a bunch of random acronyms.
Still definitely feels like that sometimes.
Oh, for sure. But they want you to understand it because when you look at that arrested embryo, or when you're deciding which culture media to use or how long to leave a dish outside the incubator, you are actively interacting with these specific molecular mechanisms.
Yeah, so our mission today is to move from the physical architecture of the cell, like the nucleus, the envelope, the pores, and dive straight into the chaotic, highly reactive, and frankly just mind-bending world of nuclear function and epigenetic regulation.
We really need to completely dismantle the idea that the nucleus is just some quiet, static library where DNA books just sit safely on shelves.
Right, it's not a museum.
Not at all. The genome is a dynamic, constantly shifting landscape. We are talking about chemical locks, physical bulldozers, invisible ink, and these crazy parental tugs of war that actually determine whether an embryo thrives or perishes right there in your lab.
I know that as embryologists, we are all very comfortable with the physical manipulations, right? Like, we know how to strip cumulus cells. We know how to do ICSI. But to truly master this field, we have to understand the molecular why behind the clinical what.
Yeah, so let's start at the absolute most fundamental unit of organization. Because before a single gene can be read to make a protein, the cellular machinery actually has to be able to physically reach it.
And for a really long time, textbooks just taught us that histones were basically these passive plastic spools that DNA just wound itself around to, avoid getting tangled up.
Yeah, just a purely structural packaging system. But that view is completely obsolete now.
Thank goodness.
Right. To understand epigenetics to this exam, you have to reframe the nucleosome, which, just as a reminder, is the DNA wrapped around that histone spool. You have to see it as a highly reactive signaling platform.
It's basically a physical switchboard. So let's look at the components of that switchboard. You have the 4 core histones, which are H2A, H2B, H3, and H4.
And 2 of each come together to form the histone octamer, which is the core of the spool.
Right, and the DNA double helix wraps roughly, 1.6 times around that core?
Exactly, 1.6 times. But the most critical part for regulation isn't the core itself. It's the N-terminal tails.
Okay, so these are the flexible protein strands that essentially just dangle off the main body of the histone, right? Like, they protrude outward away from the protected core, straight into the surrounding nuclear soup.
They do. And because they are sticking out like that, they are physically exposed and highly chemically vulnerable. The cell basically uses these protruding tails as a canvas.
So specialized enzymes are just constantly swimming by, right?
Yeah, swimming by and adding or removing tiny chemical groups to specific amino acid residues on these tails. We refer to these as post-translational modifications, or PTMs.
Because the protein has already been translated and built, and now we were just modifying it on the fly.
Precisely.
And I want to be absolutely crystal clear here for anyone studying for the ESHRE. These modifications do not alter the sequence of the A, C, T, and G bases in the DNA. The genetic code itself remains completely identical. We are just changing how the school behaves.
Yes, that is the defining feature of all epigenetics. You leave the hardware alone, and you alter the software.
I love that analogy.
These PTMs dictate the physical structure of the chromatin, and they recruit other proteins. And they pull this off through two primary mechanisms. The first one is purely physical. It's all about altering the electrostatic charge.
Okay, so think about the fundamental physics of DNA here. Deoxyribonucleic acid has a sugar phosphate backbone that carries a really strong negative charge.
Meanwhile, histones are packed with amino acids like lysine, which carry a strong positive charge.
Opposites attract. The positive spool tightly grips the negative DNA.
Like a magnet. It holds the DNA so incredibly tightly that the massive molecular machines required to transcribe a gene simply cannot wedge themselves in. The chromatin is closed.
But if a chemical modification comes along and neutralizes the positive charge on that lysine, the magnetic attraction just suddenly disappears.
Exactly. The histone loses its tight grip, the DNA physically loosens and relaxes away from the spool, and bam, the transcription machinery can finally slide in.
That brings us to the first major modification everyone needs to know for the exam, which is acetylation. This is the ultimate unlocker.
Acetylation is simply the addition of an acetyl group directly to a lysine residue on the histone tail. And the enzymes responsible for writing this mark are called histone acetyltransferases, or HATs.
HATS, and the really important ones you might see in the literature are CBP, P300, and PCAF, right?
Yeah, those are the big ones. When a HAT slaps an acetyl group onto a lysine, it neutralizes that positive charge we just talked about. The chromatin opens up. Transcription can begin.
But biological systems always have an off switch. You can't just leave it open forever. So if HATs are the writers, who are the erasers?
Those would be the histone decatylases, or HDACs.
Okay, the DACs.
Right. They come in, they rip the acetyl group off the lysine, restore the positive charge, and the DNA just snaps tightly back onto the spool, totally shutting down transcription.
Okay, I want to tie this directly to what happens in the IVF dish, because we watch oocytes mature all the time. We see them progress from the germinal vesicle stage, resume meiosis, push through M1, and eventually arrest at M2, where they just sit and wait for a sperm.
Visually, you see the polar body extrude.
Right. But molecularly, what is actually happening to the acetylation state of that oocyte's chromatin during that entire M1 to M2 journey?
It is undergoing a huge global wave of histone acetylation. The chromatin has to become incredibly relaxed and open to allow for the massive nuclear reorganization that's required for germinal vesicle breakdown and chromosome segregation.
So the oocyte is intentionally driving up HAT activity and suppressing HDAC activity.
Precisely.
So if HDACs normally remove acetylation, and we want to keep the chromatin artificially open in the lab, is this why we occasionally use HDAC inhibitors when we are trying to artificially activate an oocyte? You know, like after a failed ICSI or for specific patients with activation deficiencies?
Yes, that is the exact mechanistic rationale. When we use a compound like trichostatin A or TSA, we are deploying a chemical HDAC inhibitor. We are blocking the erasers.
So because the acetyl groups can't be removed, the oocyte's chromatin is forced to stay in a highly relaxed, hyperacetylated, totally permissive state.
You are basically chemically tricking the chromatin into adopting the open architecture that naturally occurs post-fertilization, completely bypassing the need for the sperm to trigger that cascade.
It's honestly amazing to think that just a drop of a chemical in a dish is fundamentally manipulating electrostatic charges on these microscopic spools.
It really is.
Okay, so for the exam, candidates also need to recognize the specific shorthand for these marks. It can look like a random string of letters and numbers.
Yeah, but the nomenclature is actually very logical once you see the pattern. It names a histone, then the amino acid, its position number, and finally the modification. So for example, H3K27AC. That means histone 3, lysine, which is K at position 27, is acetylated.
H3K27AC. Got it. And that specific mark tells you what?
That mark tells you that a genomic enhancer is active. Another really crucial one is H3K9AC, which generally flags an active gene promoter.
Okay, acetylation makes total sense. It neutralizes the charge, the DNA falls open. But let's look at the other major histone modification, which is methylation. Because methylation doesn't play by those same electrostatic rules, does it?
Not at all. Adding a methyl group to a lysine or an arginine residue does not neutralize the positive charge.
So if the charge doesn't change, methylation doesn't automatically cause the DNA to relax.
Right. So if it doesn't physically loosen the DNA, how does it control gene expression? Well, it acts as a highly specific docking site.
Ah, so it's like a landing pad for other proteins.
Exactly. It creates a distinct physical shape that is recognized by entirely different sets of specialized proteins, which we call readers.
Okay, and who are the writers and erasers here?
Methylation is written by histone methyltransferases, or HMTs enzymes like EZH2 and G9A, and it is erased by histone demethylase, or HDMs like LSD1.
And because methylation relies on recruiting these secondary reader proteins, its effect is totally context-dependent, isn't it?
Yes, 100%. It can be activating or deeply repressive, depending entirely on which specific glycine is methylated, and crucially whether it has 1, 2, or 3 methyl groups attached.
I can see how this gets confusing for someone studying. You really have to memorize which landing pad summons the activation crew and which one summons the lockdown crew. So what are the major activating methylation marks we need to just burn into our memory?
The absolute gold standard for an activating mark is H3K4ME3, so that's histone 3, lysine 4 trimethylation.
H3K4Me3, okay.
When you see H3K4ME3, you are looking at the promoter of a gene that is actively being transcribed. It's basically a giant neon open for business sign.
I like that. Are there any other big activating ones?
Yeah, another one is H3K36ME3, which isn't at the promoter, but it's actually found along the body of the gene itself, acting almost like runaway lights, guiding the transcription machinery along.
Okay, so H3K4ME3 is the neon sign. H3K36ME3 is the runway lights. What about the repressive marks, the ones that signal a complete lockdown?
For these, you really need to distinguish between two incredibly important ones because they represent two totally different types of silencing. The first one is H3K9AIM3.
H3K9ME3. Okay, and what does that one do?
This marks constitutive heterochromatin.
Constitutive meaning like permanent, structurally dense lockdown.
Yes. Think of the centromeres or telomeres. These are areas of the genome that the cell almost never needs to read. HCK9EP3 recruits a reader protein called HP1, which physically aggregates and just crunches the chromatin into a dense, completely inaccessible rock.
Wow, okay, and the second repressive mark?
The second one is H3K27E3, and this is the hallmark of facultative heterochromatin.
Facultative. So that implies it's conditionally silenced, right? Like a temporary lockdown.
Exactly. This mark is written by the polycomb repressive complex, specifically an enzyme we mentioned earlier called EZH2. It shuts down genes that the cell doesn't need right this second, but will absolutely need later during development.
So it's the cell's way of pausing a program without completely destroying the hard drive.
That's a great way to put it. For early embryo development, and especially for maintaining the totipotent state of the stem cells in the inner cell mass, balancing this temporary polycomb silencing is arguably the most critical epigenetic task the embryo performs.
Man, this really starts to build a bigger picture. We have acetylation physically opening doors. We have methylation routing traffic with these landing pads. But there are two other histone modifications that are highly relevant to clinical embryology, and those are phosphorylation and ubiquitination.
Let's do phosphorylation first. It's fascinating because it's usually tied directly to the physical cell cycle. Kinases add phosphate groups, and phosphatases remove them.
Okay, so what marks are we looking for here?
If you see the marks H3S10PAs or H3S28PAs globally spreading across the chromatin, it really only means one thing. The cell is entering M phase. The chromosomes are aggressively condensing to prepare for segregation.
Which is exactly why an oocyte stuck in M2 has a completely different phosphorylation profile than, say, a fertilized zygote entering interphase.
Right.
But phosphorylation also acts as an emergency alarm system, right? Like a distress signal.
Oh, it is the ultimate 911 call for the genome. I'm talking specifically about a mark called gamma H2AX.
Gamma H2AX. I've seen that in so many papers.
Right, because H2AX is a slightly different variant of the standard H2A histone. If a DNA strand suffers a double-strand break, which, by the way, is a catastrophic event for a cell within seconds, the H2AX histones surrounding that break are phosphorylated at serine 139.
And that specific phosphorylation is what we call the gamma H2AX mark.
Yes. And it spreads outward from the break site for literally millions of base pairs in both directions. It serves as this massive, screaming beacon that recruits the DNA damage response machinery like BRCA1 and ATM to come and physically stitch the DNA back together.
Think about the clinical implications of this in the lab. We know our culture media, ambient light from the microscopes, fluctuating oxygen tensions, all of these things can induce reactive oxygen species, or ROS.
And oxidative stress causes DNA double-strand breaks.
Exactly. So if an embryologist is cultivating embryos in a suboptimal environment with high oxidative stress, those embryos are likely just desperately flashing gamma H2AX marks across their entire genomes, trying to repair the damage before the next cleavage division.
And if they fail to repair it in time, the embryo either undergoes apoptosis or it arrests. That day 3 fragmentation you see so often, it is frequently the downstream result of an overwhelming gamma H2AX alarm that just couldn't be resolved.
Wow. It really makes you look at a fragmented embryo with a lot more empathy.
It really does. They're fighting a molecular war in there.
Absolutely. Okay, finally, let's briefly touch on ubiquitination. It sounds heavy.
Well, it is heavy, literally. Most of the modifications we've talked about are tiny chemical groups, just a few atoms. But ubiquitin is an entire 76 amino acid protein that gets covalently attached to the histone tail. It's a massive, extremely bulky addition.
Does it open or close the chromatin?
It depends. H2AK119EB1 is typically repressive, and it often works alongside the polycomb complex we mentioned earlier. But conversely, H2BK120EB1 actually physically wedges the chromatin open, and it is actually required to properly lay down the activating H3K4 amio3 mark.
So what you're describing with all of these acetylation, methylation, phosphorylation, ubiquitination is that no single mark acts independently.
Never. They never act alone. And that realization birthed what we call the histone code hypothesis.
Use histone code.
But the idea is that the true regulatory language of the cell isn't a single tag. It's a combination of tags on a nucleosome. It's a combinatorial code. You have writers adding marks, erasers removing them, and these specialized readers interpreting the combinations.
So like a reader with a bromodomain scans the genome looking specifically for acetylated lysines.
Exactly. And a reader with a chromodomain or a PhD finger scans for specific methylated lysines. The cell computes this incredibly complex barcode to decide exactly what to do with a region of DNA.
It is an incredibly elegant system.
Yeah.
But let's look at a physical reality here. Chemical tags and electrostatic charges are powerful, but DNA is massive, and these histone spools are really bulky.
They are.
Sometimes just changing the chemical tag isn't enough to expose a buried gene promoter. Sometimes you literally have to shove the spool entirely out of the way.
Now you're transitioning from the chemical layer to the physical layer. This is ATP-dependent chromatin remodeling.
Okay, explain this mechanism to me. Because we aren't just neutralizing a charge anymore. We are talking about literal molecular motors.
We are talking about massive, multi-subunit protein complexes that act like cellular bulldozers. They consume raw cellular energy ATP to physically alter the architecture of the nucleosome.
How do they alter it? Like, what are the actual movements?
They can do 3 main things. They can slide a histone spool further down the DNA strand, they can restructure the spool to make the DNA loop out, or they can completely eject the entire histone octamer off the DNA, creating a totally bare nucleosome-free region.
I just love the visual of a motor unjamming a zipper. If a critical developmental gene is zipped tight around a histone, the transcription machinery can't reach it no matter what chemical tags are on the tail. It needs a bulldozer to slide that spool down the track.
Exactly.
The ESHRE exam outline specifically highlights 4 major families of these ATP-dependent remodelers. Let's walk through them, starting with the most famous one, SWA-SNF.
Right. The SWA-SN family, which is also known as the Bios complex in mammals, is the heavy lifter for opening chromatin. Its primary job is to slide nucleosomes or completely evict them.
So when would the cell use SWA-SNF?
Well, when a pioneer transcription factor needs to activate a totally silenced gene, it recruits the SWA-SNF complex to physically clear the roadblocks from the promoter.
Okay, got it. Then you have the ISWI family, which seems to do the exact opposite.
ISWI acts a lot more like a measuring tape. It slides nucleosomes not to evict them, but to space them out at perfectly regular, even intervals. And regular spacing actually compacts the chromatin and makes it uniformly dense.
Why would it want to do that?
This is incredibly important right after DNA replication when the cell needs to quickly and efficiently package the newly synthesized DNA.
Makes sense. The third family is CHD. What makes it unique?
The CHD family is an amazing piece of multi-tool engineering. It possesses the ATP-dependent motor to slide nucleosomes, but it is also physically coupled to a histone deacylate and HDAC.
Oh, wow. So it brings the motor and the eraser together in one complex.
Exactly. It slides the nucleosome into a repressive position and simultaneously rips off the activating acetyl groups to chemically lock it down. It perfectly integrates physical repositioning with chemical silencing.
That is highly efficient. And the fourth family, INO80.
INO80 is the part swapper. While it's involved in DNA repair, its most unique function is histone variant exchange. Normally, you have your standard H2A histones in the core. INO80 can pry open a nucleosome, pull out a standard H2A, and wedge in a specialized variant called H2AZ.
And what does swapping that one part do?
Swapping that single part completely alters the thermodynamic stability of the nucleosome, which is highly relevant at gene promoters. It makes it easier to open later.
There is another crucial variant exchange we really need to discuss, which is central to what happens right after the sperm meets the egg. Swapping out standard histone 3 for the variant H3.3.
Yes. Incorporating H3.3 is the hallmark of highly active, uniquely flexible chromatin. When the sperm enters the oocyte, its DNA is packed impossibly tight with protamines, not histones.
Right, because the sperm has to be this tiny, streamlined missile.
Exactly. But to form a functional paternal pronucleus, those protamines have to be violently stripped out, and the oocyte's machinery rapidly deposits H3.3 in their place. This creates a hyper-accessible paternal genome, perfectly ready for the massive reprogramming events that are about to follow.
Let's ground this back in the clinical lab for our listeners. Why should an embryologist care about the SWES-NF bulldozer or the INO80 part swapper?
Because without these physical remodelers, the early embryo simply halts in its tracks. We talked about zygotic genome activation the moment the embryonic DNA actually takes over. For the DNA to activate, pioneer transcription factors must bind.
But the DNA is physically covered in nucleosomes.
Right. If the SWES-NF complexes fail to burn ATP and bulldoze those nucleosomes out of the way, the genome remains physically inaccessible. The transcription factors just bounce off. The power never turns on.
And once again, that beautiful embryo sitting in your incubator suffers a hard, irreversible developmental arrest.
Exactly. The physical mechanics of these remodelers are literally the difference between an embryo that reaches the blastocyst stage and one that degrades in a dish.
Incredible. Okay, we've spent a lot of time on the histones, the spools, the tags, the motors. But the histones are really just the packaging. Let's move to the actual code itself. Let's talk about the semi-permanent ink of the epigenome, DNA methylation.
This is huge. If histone modifications are temporary pencil marks, constantly being written and erased by the environment, DNA methylation is thick, semi-permanent ink. It is the most chemically stable epigenetic modification we know of. It is robustly maintained through decades of cell divisions.
Let's break down the actual chemistry of this ink. What exactly is a methyl mark on DNA?
It is the covalent attachment of a tiny methyl group, literally just one carbon and 3 hydrogens, to the 5 position of a cytosine ring directly on the DNA backbone.
And in mammals, this doesn't just happen anywhere on the DNA, does it?
No, it happens almost exclusively at CpG dinucleotides.
CpG, meaning a cytosine immediately followed by a guanine on the same DNA strand. And the P just stands for the phosphate backbone connecting them.
Exactly. When a cytosine in a CpG site gets methylated, it becomes 5-methylcytosine or 5-LNC. This modification is so stable and so fundamentally alters the function of the DNA that molecular biologists routinely call 5-MilC the 5th base of the mammalian genome.
The 5th base. I love that. And what does this 5th base actually do? Conceptually, we know it silences genes. But how?
By 2 distinct methods. The first is steric hindrance.
Steric hindrance, like physical blocking.
Yes. A methyl group is a physically bulky addition sticking out right into the major groove of the DNA double helix. When a transcription factor floats by trying to read a specific DNA sequence, that physical bulk literally blocks it from binding. It's like putting a thick piece of tape over a barcode. The scanner just can't read it.
Okay, that makes sense. And the second method?
The second method is active recruitment. 5-MilC serves as a massive beacon for a family of proteins called methyl CpG binding domain proteins, or MBDs.
Okay, think of proteins like MESI-P2.
Yes, exactly. These readers specifically hunt for methylated DNA. When they bind, they act as massive anchors, dragging along enormous repressive complexes, including HDACs and those chromatin remodelers we talked about, that completely shut down and compact the surrounding area.
So this ink is incredibly powerful. But if it's so stable, how does a cell ensure it gets passed down when it divides? Like, a skin cell has to make another skin cell, which means the daughter cell needs the exact same methylation pattern as the parent. What is the copying mechanism?
To understand this, you have to divide the enzymes that handle the ink, the DNA methyltransferase or DNMTs, into 2 categories. You have the writers of new ink and the copiers of old ink.
Let's start with the writers.
The writers are the de novo methyltransferases DNMT3A and DNMT3B, supported by a structurally similar but catalytically inactive cofactor called DNMT3L. They are responsible for laying down entirely new patterns of methylation during gametogenesis and early embryonic lineage specification.
But once the pattern is established, The cell relies on a completely different enzyme to copy it during division, right?
Yes, the maintenance methyltransferase, DNMT1, and its mechanism is just beautifully elegant. During the S phase of the cell cycle, the DNA double helix unzips to replicate. The original parental strand still has all its 5' C marks intact, but the newly synthesized daughter strand is completely bare.
So for a brief window, you have this hybrid DNA molecule. One strand is heavily inked, and the other is a totally blank slate.
We call that hemimethylated DNA. Now, DNMT1, aided by a crucial partner protein called UHRF1, specifically recognizes these hemimethylated CpG sites. It binds to the hafting site, reads the parent strand, and chemically stamps a matching methyl group onto the cytosine of the bare daughter strand.
Wow. So it is an obsessive copying machine that ensures epigenetic fidelity across millions of cell divisions.
Precisely.
There's a structural concept related to this that I know ESHRE loves to test on, CpG islands.
Right, if CpG sites are the specific locations of the ink, CpG islands are unique architectural features. They are stretches of DNA, typically hundreds of base pairs long, that are incredibly dense with CpG dinucleotides. Curiously, they're usually found right at the promoters of housekeeping genes and critical developmental regulators.
And the rule of thumb is that in a healthy, normal cell, CpG islands at active promoters are strictly kept completely unmethylated.
Right, they are heavily protected from the DNMTs. But in diseases, most notably in cancer, the epigenome breaks down. You get aberrant hypermethylation of these CpG islands, which permanently turns off tumor suppressor genes.
And in the context of IVF, the clinical fear has always been whether our artificial culture systems might occasionally trigger this exact same aberrant hypermethylation at critical developmental genes.
Which is a very valid concern.
Okay, so the ink is incredibly stable, and DNMT1 is ruthlessly copying it every time the cell divides. But for reproduction to work, the embryo cannot just keep the parent's somatic methylation marks. It has to wipe the slate clean to achieve totipotency, the ability to become any cell type at all. How do you erase semi-permanent ink?
The erasure process is honestly one of the most drastic and dangerous events in cellular biology. The cell executes a massive whiteout process using two completely different strategies: passive erasure and active erasure.
Passive erasure sounds like the cell just gets lazy.
That's essentially what it is. Passive demethylation relies purely on dilution. The zygote enters a period of extraordinarily rapid cleavage divisions. If the cell deliberately excludes DNMT1 from the nucleus or simply downregulates its expression, the newly synthesized DNA strands just don't get copied.
Oh, I see. So with the first division, the overall methylation drops to 50%.
Yes. The next division, 25%. The next, 12.5%. Through sheer dilution over several days, the ink slowly fades away to nothing.
But dilution takes time. Sometimes the cell needs the ink gone right now, immediately after fertilization.
And that requires active demethylation. This isn't passive at all. This is an aggressive, chemically violent attack on the methyl group, driven by the 1011 translocation enzymes, the TTT enzymes.
Specifically, TT1, TT2, and TET3.
Yes.
Walk us through the chemistry of a TET enzyme, because you can't just snip the methyl group off a cytosine, right? The carbon bond is way too strong.
Exactly. The cell cannot physically break that bond directly. So instead, the TET-E enzymes use a strategy of progressive chemical rusting. They aggressively oxidize the methyl group in a stepwise fashion. First, a TE enzyme oxidizes 5-methylcytosine into 5-hydroxymethylcytosine, or 5-LDFC.
Let me stop you there for a second, because 5-LMSC is more than just a stepping stone, isn't it? It actually acts as its own functional mark.
It does. 5-LFEMC fundamentally alters the structure of the DNA, and it recruits an entirely different set of reader proteins compared to 5-LC. But if the ultimate goal is complete erasure, the TET-E enzymes just keep going. They oxidize the 5-LVMC into 5-formal cytosine, or 5FC.
And then?
And then they hit it a third time, oxidizing 5-SC into 5-carboxylcytosine, or 5-KC.
So it literally rusts the methyl group 3 separate times until it is chemically mangled. What happens to the 5-KC?
Well, at this point, the cell has finally disguised the methyl group as a corrupted, damaged base. And this triggers the base excision repair machinery. An enzyme called thymine DNA glycosylase, or TDG, scans the DNA, recognizes 5-FC and 5-KC as damaged bases, and literally cuts the entire modified cytosine right out of the DNA backbone.
It just chops it out.
It leaves a temporary hole, which is quickly filled with a fresh, completely unmethylated cytosine by DNA repair polymerases. The ink isn't just covered up. The rusted metal is surgically excised and replaced.
It is stunningly complex, and this massive wave of active erasure happens immediately after fertilization, sweeping across the genome to restore totipotency. But there is a second massive wave of erasure that is critical for embryologists to understand, which happens much later, in the primordial germ cells.
Yes. Once the embryo has implanted and is developing into a fetus, it sets aside a tiny population of cells that will eventually become its own sperm or
Yes.
eggs, the primordial germ cells or PGCs.
And by the time these cells are allocated, they have already acquired a bunch of somatic methylation marks from early fetal development.
Right. To become true germ cells, they need a total factory reset.
The ultimate reboot.
It is the most profound epigenetic erasure in mammalian biology. Driven by TEP-T1 and TEP-T2, combined with passive dilution, almost the entire methylome is obliterated. And here's the most critical part for the exam. This is the exact and only moment when the genomic imprints inherited from the previous generation are finally erased.
Wow. So the slate is wiped entirely clean so that this new developing fetus can establish its own sex-specific imprints during its own gametogenesis.
Precise.
That perfectly sets up our next major section, because we just said the entire genome gets wiped right after fertilization. But during that first chaotic wave of TET-mediated erasure, a tiny fraction of the genome absolutely must hold onto its ink. If it loses it, the embryo develops catastrophic disorders. I'm talking about genomic imprinting.
Genomic imprinting completely breaks the fundamental rules of Mendelian genetics we all learned in high school. Usually, you inherit two functional copies of a gene, one from your mother, one from your father.
Yeah.
If one is mutated, the other provides a backup.
Right, standard genetics.
But for about 100 to 200 specific, highly critical genes in humans, expression is monoallelic and strictly parent of origin dependent. The cell actively silences one parent's allele and relies 100% on the other parent's allele.
And the embryo knows which is which, not because the DNA sequence is different, but because of the epigenetic ink.
Correct. The memory of where the gene came from is encoded by localized DNA methylation at specific regulatory sequences called imprinting control regions, or ICRs. Sometimes you'll see them called differentially methylated regions, or DMRs.
So at an imprinted locus, one parent's allele arrives in the zygote heavily methylated and silenced, and the other parent's allele arrives completely bare and active.
Exactly.
But let me play the skeptic here. We just established that right after fertilization, the egg cytoplasm is packed with TET enzymes that are aggressively hunting down and rusting away every methyl group they can find. How on earth do these specific imprinting marks survive that chemical firestorm?
It's a great question. They survive because the cell deploys elite molecular bodyguards specifically to protect them. The oovocyte provides specialized proteins like the DPPA3 complex, which is also known as STELA, and the ZFP57 TRIM28 complex.
STELA and TRIM28.
Yes, these proteins physically bind to the imprinted ICRs. They form an impenetrable shield over the methylated DNA, physically blocking the T enzymes from reaching the cytosines and protecting them from passive dilution by actively recruiting DNMT1. They ensure that critical imprints survive the reprogramming phase and make it safely into the developing blastocyst.
The ESHRE board definitely expects candidates to recognize the specific imprinted gene clusters. Let's list the heavy hitters.
The most famous cluster is located on chromosome 11, specifically at position 11p15.5. This region is an absolute master controller of fetal growth. It houses the IGF-2 and H19 locus.
Okay, so IGF-2 is a potent growth factor, and it is expressed exclusively from the paternal allele.
Right, and H19 is a non-coding RNA that suppresses growth, and it is expressed exclusively from the maternal allele. Also in that exact same 11p15.5 cluster is KCNQ1 and CDKN1C, which are maternally expressed and control cell cycle progression.
Okay, so that's the chromosome 11 cluster and the other major chromosomal region.
Chromosome 15, specifically 15q11q13. This region houses the SNRPN and SNORD cluster, which is strictly paternally expressed, and the UBE3A gene, which is maternally expressed in the brain.
You also see the IGF-2R gene mentioned in animal models a lot, right?
Yes, IGF-2R is a classic maternally expressed imprint in mice that acts basically as a sink to clear away excess IGF-2 growth factor.
You know, whenever I review imprinting, I always have to ask, from an evolutionary standpoint, why does this system even exist? It just seems incredibly reckless. Why would a developing embryo intentionally and permanently turn off a perfectly good backup copy of a vital growth gene? If the one active copy gets mutated, you have literally no safety net.
It is incredibly fragile.
What?
The prevailing evolutionary explanation for why mammals evolved this system is David Haig's genomic conflict hypothesis, which is sometimes called the kinship theory. It posits that imprinting is the molecular result of an evolutionary tug-of-war over maternal resources during pregnancy.
A battle of the sexes playing out at the molecular level.
Exactly. From a purely evolutionary perspective, the father's genes want the offspring to grow as large and robust as possible to ensure survival, regardless of the physical cost to the mother. Therefore, paternally expressed imprinted genes, like IGF-2, act as aggressive growth promoters, extracting maximum nutrients across the placenta.
But the mother's evolutionary priority is different. She wants the offspring to survive, sure, but she also needs to survive the pregnancy herself so she can reproduce again in the future.
Precisely. If the fetus grows too aggressively, it could kill her. So maternally expressed imprinted genes, like H19 or IGF-2R, act as growth restrictors. They hit the brakes, counteracting the paternal growth signals to preserve maternal fitness. The healthy human embryo represents the exact precarious stalemate of this tug-of-war.
But what happens when that fragile stalemate is broken, especially in the context of the IVF lab? If the bodyguards fail or the environment causes the methylation ink to smudge, you lose that parent of origin dosage. And that leads us to imprinting disorders.
When the balance tips, whether from aberrant methylation, a chromosomal deletion, or uniparental disomy, where you accidentally inherit two copies of a chromosome from the same parent, the results are severe developmental syndromes.
Let's start with the disorders mapping to that growth cluster on chromosome 11p15.5, specifically Beckwith-Wiedemann syndrome, or BWS.
BWS is the classic overgrowth syndrome. The maternal brakes fail, or the paternal accelerators are doubled. Patients present with macroglossia, which is an enlarged tongue, massive organomegaly, umbilical hernias, and a significantly increased risk of childhood cancers like Wilms tumor.
And molecularly, what causes it?
It is most frequently caused by a loss of maternal methylation at the KCNQ1 ICR, causing a massive dysregulation of the entire growth cluster.
And what is the specific relevance to the clinical embryologist?
This is the critical clinical stat for the exam. Children conceived through assisted reproductive technologies, specifically IVF and ICSI, show an approximately six-fold increased relative risk of developing Beckwith-Wiedemann syndrome compared to naturally conceived children.
Sixfold. Wow.
Yeah, the epidemiological evidence strongly suggests that our in vitro culture conditions or stimulation protocols are stressing the epigenome and occasionally causing that maternal methylation mark to drop.
Okay, what about the exact opposite phenotype, the undergrowth syndrome?
That would be Silver-Russell syndrome, or SRS. It is characterized by severe intrauterine and postnatal growth restriction, a distinctive triangular face, body asymmetry, and relative macrocephaly. It is the molecular inverse of BWS.
So it's caused by a paternal defect.
Yes, it is frequently caused by the loss of paternal methylation at the H19 IGF-2 ICR on chromosome 11, meaning the growth promoters are silenced and the brakes are locked on. SRS also shows an elevated risk in the RT population, roughly a 2 to 4-fold increase.
Now let's move to chromosome 15, because this provides the ultimate textbook demonstration of parent of origin effects. Two completely different, devastating syndromes caused by defects at the exact same physical location. 15q11q13, Angelman syndrome versus Prader-Willi syndrome.
It is an absolute masterclass in epigenetics. If a developing fetus loses the maternal expression of the UBE3A gene in this cluster, perhaps the mother's allele was deleted, or the paternal allele was duplicated through uniparental disomy, or an imprinting center defect erased the maternal mark, the child develops Angelman syndrome.
And what are the symptoms of Angelman?
These patients suffer from severe intellectual disability, an almost complete absence of speech, frequent seizures, and a characteristically happy, easily excitable demeanor, which is why it was historically, though insensitively, called happy puppet syndrome.
But if the defect hits the exact same physical location on the paternal chromosome,
You get a completely different clinical reality, Prader-Willi syndrome. PWS results from the loss of paternal gene expression at the SNRPN cluster on 15q11q13. These infants present with profound neonatal hypotonia floppy baby syndrome.
and then that transitions into hyperphagia later, right?
Yes, as they grow, they develop extreme, uncontrollable hyperphagia, an insatiable appetite that leads to life-threatening severe obesity. They also suffer from hypogonadism and intellectual disability. Same DNA sequence, totally different epigenetic origin, totally different disease.
I have to pause here and just channel every clinical embryologist listening. When we hear these statistics, a six-fold increased risk of BWS, a four-fold increase in SRS, the immediate terrifying implication is that our culture media, our incubators, or our ICSI needles are actively causing epigenetic brain damage to these embryos. Is that the undisputed truth? Or is it possible that a patient seeking IVF already has underlying inherent epigenetic flaws driving their infertility?
It is an essential pushback. And the reality is a deeply intertwined mix of both patient-derived risk and laboratory-derived risk. It is almost impossible to cleanly separate them. Let's look at the patient side first. We know definitively that severe male factor infertility oligosospermia, teratospermia, is highly correlated with abnormal DNA methylation patterns at imprinted loci in the sperm itself.
So the sperm arrives at the wrong ink already.
Yes. In natural conception, a sperm with massive epigenetic defects usually fails the grueling physical marathon through the female reproductive tract. But in the lab, we use ICSI. We completely bypass natural selection. An embryologist picks a morphologically normal-looking sperm and injects it. But morphology does not equal epigenetic competence.
So we might be manually introducing a pre-existing imprinting error into the oocyte.
Exactly. That is patient-derived risk, facilitated by technology.
But the laboratory environment isn't exactly innocent either, is it?
Not at all. We have overwhelming evidence from animal models that the physical environment directly alters the epigenetic machinery. We will evaluate those specific RP risks in detail shortly. But the main takeaway is that the baseline infertility provides the epigenetic vulnerability, and the artificial stress of the laboratory provides the trigger.
Speaking of the laboratory timeline, let's zoom back in on the embryo itself in the days following fertilization. We established that the imprinted genes are the rare exceptions. They keep their parental ink. But the rest of the genome has to be wiped clean and turned on. How does an embryo transition from a silent, single-cell zygote into a transcriptionally active, multicellular organism? Let's explore zygotic genome activation.
To understand ZGA, you have to realize that a day one zygote is entirely dependent on maternal life support. Every single protein functioning in that zygote, every structural filament, every enzyme was either prepackaged into the egg cytoplasm during its time in the ovary or translated from a massive stockpile of maternal mRNA transcripts the oocyte hoarded.
So the newly combined DNA of the sperm and egg, the embryonic genome, is just sitting there, fully formed but totally silent.
It's like moving into a fully furnished, highly complex factory before the main power grid has been connected. The backup generators are running, but fuel is limited.
That is precisely the situation. The embryo has a ticking clock. The maternal supplies are degrading. The power grid has to be switched on before the backup generators fail or the embryo dies.
That switch-on event is zygotic genome activation. In mammals, it generally occurs in two distinct waves. There is a minor wave of ZGA that happens very early, right around the one-cell or two-cell stage, involving just a handful of genes.
But the main event, the major ZGA, happens a bit later.
In humans, the major ZGA occurs at the 4 to 8 cell stage. This corresponds exactly to day 3 of development in the IVF lab. This is a massive, genome-wide burst of transcription. The embryonic DNA finally wakes up, starts pumping out its own mRNA, and officially takes control of its own developmental destiny. It cuts the molecular umbilical cord to the oocyte.
But a genome doesn't just spontaneously wake up. We spent the first half of this deep dive discussing how tightly locked down DNA is. What prerequisites must be cleared for major ZGA to occur?
It requires a monumental orchestration of all the remodeling tools we discussed. First, the paternal genome must be stripped of its tight cotamine packaging and loaded with H3.3 histones. Then, both genomes must undergo massive demethylation to erase the repressive ink. Repressive histone marks must be removed.
And crucially, even after the ink is gone, the chromatin is still physically closed. You need pioneer transcription factors to force the doors open.
Right. In humans, the star pioneer factor is a protein called DUX4.
DUX4. It acts as the ultimate molecular crowbar, doesn't it?
It really does. It has the unique ability to bind to closed, nucleus-dense heterochromatin. It binds, recruits those Swiss SM bulldozers, forces the chromatin open, and creates a physical beachhead so the rest of the standard transcription machinery RNA polymerase can land and begin transcribing the massive burst of embryonic genes.
And while this frantic building up of embryonic machinery is happening, the old maternal machinery is being intentionally demolished.
That concurrent process is the maternal to zygotic transition, or MZT. As the embryonic genome roars to life, it actually transcribes specific microRNAs and decay enzymes whose sole purpose is to hunt down and destroy the remaining maternal mRNA transcripts. The oocyte's legacy is systematically deadenylated and degraded to ensure the embryonic genome has absolute, undisputed control.
Let's bring this back to the embryologist staring down the microscope. We mentioned the tragedy of the day 3 arrest at the start of the deep dive. When an embryologist sees an 8-cell embryo that looked perfect yesterday, but today the blastomeres are totally stalled, what are they witnessing at a molecular level?
They are looking at a failed MZT. The embryo successfully executed the degradation part. It burned through the maternal supplies and destroyed the maternal mRNA. But the activation part, the ZGA, failed. Perhaps DUX4 couldn't pry the chromatin open, perhaps the SWES-NF motor stalled, perhaps the demethylation didn't finish, the power grid never turned on.
So the embryo is sitting in the dark, out of fuel, and it just arrests. It is the ultimate epigenetic failure.
Exactly. It really changes how you look at the grading system. You aren't just grading cell size or symmetry, you are literally grading the success of a massive molecular handover.
Now, to achieve that totipotent state for ZGA, the embryo has to treat the incoming sperm and egg genomes in completely different ways in the hours right after fertilization. Let's delve into post-fertilization reprogramming.
It is a tale of two genomes experiencing two completely different timelines inside the exact same cytoplasm. Let's look at the paternal genome first. The sperm arrives as an incredibly specialized, hypercompacted, terminally differentiated missile. It has to be rapidly converted back into a flexible totipotent state. It is a violent sprint.
The outline calls it the paternal race.
Exactly. Within hours of the sperm entering the oocyte, even before the pronuclei fuse, the paternal DNA is aggressively attacked by the active demethylation machinery. The oocyte cytoplasm is highly concentrated with TET-T3 enzymes.
And TET-T3 just swarms the paternal pronucleus and launches a massive oxidation campaign, rapidly converting the 5-milli-C ink into 5-ECC and beyond.
Right. Concurrently, the protamines are ripped out and replaced by flexible H3.3 histones. Activating acetylation marks flood the chromatin. In mere hours, the paternal genome is forced open.
But the maternal genome, sitting just microns away in the female pronucleus, is largely spared from this TET-T3 assault. Why?
Because of the bodyguards we mentioned during imprinting, the maternal genome is saturated with DBPA3, or STELA. STELA physically shields the maternal chromatin, blinding the TT3 enzymes to its presence. Because it is protected from active, violent erasure, the maternal genome must undergo a marathon of passive erasure.
So it slowly dilutes its methylation over the next several days through successive cleavage divisions without DNMT1 maintenance.
Yes. So if an embryologist could use a molecular scanner to look at a day 1 zygote with two co-nuclei, they would see two fundamentally different epigenetic landscapes. The male pronucleus is a frenzy of active TE3 oxidation and histone swapping. The female pronucleus is calm, protected, and heavily methylated.
They start far apart, but they're both marching toward the exact same destination. Over the next 4 to 5 days, as the embryo cleaves and forms a morula and then a blastocyst, the genome-wide methylation levels of both sets of DNA just plummet.
By the time you have a fully expanded blastocyst, global methylation hits a nadir, its absolute lowest point of around 20 to 30%.
A completely blank slate, save for the imprinted region.
Yes, and from that blastocyst nadir, as the cells begin to differentiate into the inner cell mass, or the trophectoderm, the de novo methyltransferases DNMT3A and 3B finally switch back on and start writing new lineage-specific ink.
Amidst all this invisible genome-wide chaos, the TET-E oxidation, the protamine stripping, the DUX4 crowbars, there is one very distinct, highly visible structural change that occurs in the nucleus. And it is something embryologists rely on heavily for clinical grading. Let's talk about the nucleolus, ribosome biogenesis, and NPB scoring.
The nucleolus is a massive, dense structure inside the nucleus. It is the largest nuclear body, easily visible under a standard inverted microscope. What's totally unique about the nucleolus compared to other cellular organelles is that it has no membrane. It isn't a balloon. It forms dynamically through a biophysical process called liquid phase separation.
Phase separation. Think of a lava lamp or, shaking up oil and vinegar in a salad dressing. The molecules naturally aggregate into distinct droplets based on their biophysical properties.
Precisely. The nucleolus condenses out of the nuclear soup around very specific locations on the chromosomes. These locations contain hundreds of repeating copies of ribosomal DNA, or rDNA.
And in humans, these rDNA clusters are located on the short, stalk-like arms of the 5 acrocentric chromosomes, 13, 14, 15, 21, and 22. We call these clusters nucleolus organizing regions.
Right. And when you look closely at a functional nucleolus using electron microscopy, it's not just a uniform blob. It has a highly specific internal architecture. It has 3 distinct concentric zones that map directly to its assembly line function.
Okay, let's break down the zones. What's the innermost one?
The innermost zone is the fibrillar center, or FC. This is where the rDNA genes actually sit and where the transcription enzyme RNA polymerase I docks. Surrounding that is the dense fibrillar component, or DSC.
And what happens in the DFC?
This zone is marked by a protein called fibrillarin, and it is where the newly minted RNA transcript undergoes its very first processing steps.
In the outer layer.
The granular component, or GC, marked by the protein nucleophosmin, or NPM1. This outer layer is where the late-stage processing happens and where the actual ribosomal subunits are physically assembled before being shipped out of the nucleus.
Because that is the entire purpose of this organelle. It is literally a ribosome factory.
Exactly. RNA polymerase I transcribes the rDNA into a massive, continuous 45S pre-ribosomal RNA molecule. As that massive molecule moves outward through the zones, it is chopped up and heavily modified to produce the 18S rRNA for the small ribosomal subunit and the 5.8S and 28S RNAs for the large subunit. The nucleus is building the machines that will eventually build every protein in the cell.
Now, let's track the lifespan of this factory during reproduction. In a maturing oocyte at the germinal vesicle stage, the nucleolus is massive.
It is working in absolute overdrive. The oocyte is frantically transcribing ribosomes to build up that massive maternal stockpile we talked about, preparing for the days of silence after fertilization. But when the oocyte resumes meiosis, that massive nucleolus completely disassembles and vanishes.
And here is the clinical connection. When fertilization occurs and the male and female pronuclei form, we don't see true nucleoli return immediately. We see nucleolar precursor bodies, or NPBs.
NPBs are not functional ribosome factories, they are transcriptionally silent. Because major ZGA hasn't happened yet, the embryonic rDNA is dormant. NPBs are essentially dense protein holding pens. They gather all the nucleolar machinery, the RNA polymerases, the fibrillarin, and hold them in a phase-separated droplet waiting for the power grid to turn on.
And when ZGA finally hits on day 3,
the rDNA activates, it pierces the NPB, and a true functional nucleolus forms.
Every clinical embryologist knows the Z-scoring system or the pronuclear morphology assessments. We look at the zygote on day 1, roughly 16 to 18 hours post-insemination. We count the NPBs in the male pronucleus. We count them in the female pronucleus. We look at whether they are equal in number, whether they are symmetrical in size, and whether they have polarized meaning lined up along the junction where the two pronuclei meet. Why does the physical symmetry of these silent droplets predict whether the embryo will make it to a blastocyst?
Because the NPBs are a macroscopic readout of a microscopic epigenetic reality. Remember, NPBs form around specific regions of chromatin. If the paternal genome and the maternal genome are successfully aligning their epigenetic reprogramming, if the TT3 oxidation and the passive demethylation are proceeding synchronously, the physical architecture of the chromatin reorganizes symmetrically, dragging the NPBs into a polarized, matched arrangement.
So if an embryologist sees highly asymmetrical NPBs, say 7 tiny scattered dots in the male pronucleus and 3 large polarized ones in the female,
they are looking at severe developmental asynchrony. One genome's chromatin is behaving differently than the other. The epigenetic reprogramming is disjointed, and an embryo with disjointed reprogramming has a vastly higher chance of failing ZGA and arresting on day 3. NPB scoring isn't just about counting dots. It is a direct, non-invasive assessment of epigenetic health.
This brings us to our final overarching synthesis. We have built an incredibly complex molecular machine today, and now we have to critically evaluate how our daily actions in the IVF lab might be throwing wrenches into those gears. Let's do a critical evaluation of RT-associated epigenetic risks. The embryo is undergoing the most vulnerable, massive epigenetic remodeling of its entire life cycle, and it is doing it while sitting in a plastic dish bathed in artificial media.
The temporal overlap is the danger. We're applying environmental stress at the exact moment the epigenome is most malleable. Let's trace the clinical pathway, starting before the oocyte even leaves the body. Controlled ovarian stimulation, or COS.
The foundation of IVF. We pump the patient with supraphysiological doses of gonadotropins to rescue follicles that were destined to die.
And by doing so, we heavily disrupt the natural timing of folliculogenesis. Think back to the primordial germ cells. Oocytes are actively writing and reestablishing their maternal imprints, laying down that permanent DNMT ink during their growth phase in the follicle. By artificially rushing that growth phase with high-dose FSH, or by rescuing an oocyte that was biologically flagged for atresia due to poor competence, we risk interrupting that delicate inking process.
Do we have actual evidence that COS causes errors?
Yes. Multiple studies looking at oocytes retrieved from heavily stimulated cycles show subtle but distinct methylation losses at crucial maternally imprinted genes, like KCNQ1 and SNRPN, compared to oocytes retrieved in natural cycles. The drugs themselves aren't mutating the DNA, but they are altering the biological time frame the enzymes need to work.
Okay, then we retrieve the eggs and place them into in vitro culture. We try to mimic the fallopian tube, but a plastic dish is a poor substitute. How exactly does culture media alter the epigenome?
Through direct metabolic linkage. The methyl groups that DNMTs use to ink the DNA don't appear out of nowhere. They are synthesized by the cell's metabolic pathways, specifically the one-carbon metabolism cycle. This cycle requires precursors like methionine, folate, and specific B vitamins. The precise amino acid formulation of your culture media dictates the availability of those methyl donors.
Furthermore, suboptimal pH or high oxygen tension generates reactive oxygen species, which, as we discussed, trigger gamma H2AX alarms and can physically inhibit TE enzyme activity.
We have glaring proof of this from veterinary IVF, don't we?
Oh, absolutely. Decades ago, when agricultural scientists were culturing sheep and cattle embryos in suboptimal media containing heavy serum, they created a massive epigenetic disaster known as large offspring syndrome, or LOS. The calves and lambs were born catastrophically overgrown. When scientists looked at the molecular cause, they found a complete loss of maternal imprinting at the IGF2R locus, the exact gene responsible for clearing away excess growth factor. The culture media directly broke the imprinting mechanism.
Now, obviously, human cultured media has improved drastically since then. We don't see a direct equivalent of LOS in humans, but we do see subtle birth weight variances. What about the fertilization method, ICSI? We touched on this earlier, but let's synthesize it.
ICSI is the ultimate confounding variable. By manually selecting a sperm, we bypass the female reproductive tract's rigorous epigenetic quality control. We force fertilization using sperm from men with severe oligozoospermia or teratozoospermia. We know definitively that these compromised sperm have vastly higher rates of imprinting errors, particularly at the paternal H19IGF2 cluster. So ICSI absolutely increases the epigenetic risk profile of the resulting embryo, primarily by transmitting existing paternal errors that nature would have filtered out.
There is one final layer of epigenetic architecture that is highly vulnerable in the lab, happening right as the embryo reaches the blastocyst stage. As the embryo prepares to implant, it makes its first major life choice: lineage specification. It divides into the inner cell mass, which becomes the fetus, and the trophectoderm, which becomes the placenta. This isn't just about turning genes on and off. It's about 3D space.
It's about large-scale nuclear folding. The genome organizes itself into massive physical territories called A and B compartments. The A compartment consists of open, highly active euchromatin floating near the center of the nucleus. The B compartment consists of dense, locked-down heterochromatin, which is often physically pushed to the very edge of the nucleus and tethered to the nuclear lamina.
How does this compartment switching drive lineage?
The cells of the inner cell mass have to remain pluripotent. They need access to thousands of developmental pathways, so their genome is maintained overwhelmingly in the open A compartment. But as the outer cells commit to becoming trophectoderm, they don't need those fetal pathways anymore. They systematically take huge sections of their DNA, condense it, and physically shove it into the B compartment, permanently locking it to the nuclear wall.
And if an embryo has been sitting in suboptimal media, struggling with pH shifts, struggling with oxidative stress, fighting to repair DNA damage,
it lacks the cellular energy and the molecular precision to execute this 3D compartment switching correctly. The trophectoderm fails to properly silence the pluripotent genes, or the ICM accidentally locks away critical developmental genes into the B compartment. The lineage allocation becomes biased or ambiguous. Visually, you might see a blastocyst with a beautiful trophectoderm, but an inexplicably tiny, poor-quality inner cell mass. That is the macroscopic result of a microscopic failure in 3D chromatin folding.
Okay, we have outlined a terrifying gauntlet. We've discussed COS disrupting methylation timing, ICSI passing on sperm errors, culture media altering methyl donors, and oxidative stress ruining 3D folding. For the clinical embryologist listening, who is now terrified to even look at an incubator, give us the balanced, evidence-based reality check. How big is the actual clinical risk to a human baby?
The reality check must be rooted in absolute numbers, not relative risk. Yes, earlier we cited that the relative risk of an imprinting disorder like Beckwith-Wiedemann syndrome is increased up to 6-fold in the RT population. That sounds terrifying, but you must look at the absolute baseline. The baseline rate of BWS in the naturally conceiving population is astronomically low, roughly 1 in 15,000 births.
So if you multiply a tiny fraction by 6,
You still get a very tiny fraction. The absolute risk of an imprinting disorder in a child conceived through IVF or ICSI is approximately 1 to 2 out of every 10,000 births.
meaning that 9,999 998 out of 10,000 children born from RT are totally free from these specific imprinting disorders.
Exactly. The vast, overwhelming majority of human embryos possess an incredible epigenetic resilience. They correct errors, they adapt, and they develop into perfectly healthy children. The epigenetic signal we are seeing in the epidemiological data is a real biological warning light. It proves that our interventions have consequences. But it should absolutely not be a cause for clinical panic or patient terror.
How should this knowledge change the daily practice of an embryologist?
It should serve as the ultimate motivation to pursue absolute perfection in the lab. It reinforces exactly why we obsess over temperature recovery times when a door is opened. It explains why we strictly regulate pH, why we move to low oxygen tension incubators to reduce ROS, and why we rigorously demand extensive quality control for our culture media. We do these things to minimize the environmental pressure on a highly vulnerable epigenetic machine. When counseling patients, we acknowledge the slight increase in risk honestly, but we contextualize it with the overwhelming likelihood of a healthy outcome.
What a profound journey this has been. Let's recap the molecular landscape we've traversed. We started with the physical spools of the nucleosome, decoding how chemical tags, the acetylation unlocking the DNA, the complex routing of methylation, the gamma H2AX alarms, create the histone code. We visualized the sheer physical force of ATP-dependent remodelers like Swiss EDF bulldozing those spools aside. We explored the permanent ink of DNA methylation, laid down by DNMTs and violently rusted away by the TET enzymes in a massive wave of erasure. We navigated the fragile, evolutionary tug-of-war of genomic imprinting, seeing how bodyguards protect vital genes and how failures lead to disorders like BWS and Angelman syndrome. We tracked the high-stakes life-or-death moment of the zygotic genome activation, recognizing a day 3 arrest not as bad luck, but as a failed molecular handover. And we tied the visual symmetry of nuclear precursor bodies directly to the invisible epigenetic reprogramming happening inside the pronuclei.
It is a staggering amount of molecular machinery. The acronyms can be overwhelming: HATs, HDACs, TTE, DNMTs, GUX4, but I urge every candidate, don't just memorize the letters, visualize the machines, visualize the bulldozers and the chemical ink, because every single piece of this machinery is operating right beneath your fingertips when you are working at the ICSI rig.
Which leads me to a final, profound concept to leave you with. In the laboratory, you are constantly making tiny environmental adjustments. You tweak the CO2 to balance the pH of a dish by 1/100 of a point. You select a specific media brand. When you do these things, you are not merely a caretaker acting as a life support system for a collection of cells. You're acting as an environmental epigenetic writer. The physical parameters of your laboratory are actively feeding into a molecular symphony, shifting the balance of acetylation, altering the availability of methyl donors, influencing the timing of chromatin remodeling. The care you take in the lab doesn't just keep an embryo alive. It helps draft a molecular blueprint that will echo through the lifetime of that human being and potentially into the generation that follows.
It truly changes the weight of the profession. The DNA sequence is the hardware, but the environment you meticulously maintain in the IVF lab provides the exact conditions needed to program the software.
To every clinical embryologist preparing for the ESHRE exam, we wish you the absolute best of luck. Keep digging past the surface, keep asking why the mechanisms work, and never look at a day 3 arresting embryo or a subtle shift in culture pH the exact same way again.
Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.